Perfect. Thank you, Gabriela. Welcome, everybody. This is our activity called DNA for Dinner. It's part of the Bay Area Science Festival, which is held annually. And we are all from the CLEAR project, which is Communication, Literacy, Education, and Agricultural Research. It's part of the Plant Biology and Microbiology Department at UC Berkeley. And we're all graduate students. My name is Enrico, first-year graduate students in Plant Biology. With me is Gabriela.
Yeah, hi everyone. Like Enrico said, I'm Gabriela. I am a biologist, and so I study photosynthesis at University of Berkeley as well.
And with me is also Matthew.
Hi everyone, I'm a first-year graduate student at Berkeley as well, and I study plants, and in particular the interactions between bacteria and soybeans and tomato.
Thank you, Matthew. And for today's activity, what we'll be doing, we'll be extracting DNA from fruits of strawberries. So to get started, Gabriela has an introduction for us. So why don't you take it away?
Hey everyone, so I just want to say welcome. I'm really excited that you're all here. So as a biologist, and as Matthew and Enrico can attest to, we're really interested in how life works. And as you can see in this slide in front of you, life is pretty diverse. There's lots of things that are alive. You and I are alive, our pets are alive, and that life comes in diversity all around the planet in forms of fungi, animals in the ocean, and even tiny little organisms like bacteria. And the fact that we are all alive, we are all living organisms, that means we actually have a lot in common. And one of those main things that we have in common is that we're made up of the same units called cells. So cells are what we call the unit of life. And just as the diversity that we saw on the first slide, that diversity in cells is reflected in different organisms. So here we are looking at human skin cells. We're made up of trillions and trillions of cells. In the middle, you can see plant cell, which is unsurprisingly really green. And then there are even organisms that are only made up of one single tiny cell called a unicellular organism, which all bacteria belong to.
Great. Thank you, Gabriela. And so you may be wondering, so how do cells know what to do? Cells, as Gabriela has said, have a lot of functions and can do a lot of different things to shape the great diversity of life on the planet. Well, to know what to do, cells need to follow instructions. There is a set of directions that, everybody has to follow to do any procedure. If you want to play games like Uno, you need to know the instructions of the game. You need to know the rules of the game. If you want to build Legos, you follow the instructions that come with the game. And architects to build plans, they have to follow instructions. So what are the instructions that cells have to follow? Well, if we can see, next slide. The instructions that you have been interacting with your entire life are written in a language, and they are written with letters of the alphabet. In English, there are a certain amount of characters in the alphabet, and then different alphabets have different characters. You can see in Spanish, there are more characters as an N with a tilde. And completely different cultures have other ways of expressing their information. Here you see Chinese characters to express information. Even machines have their own way of conveying information and sharing it to know what to do. The binary code is in the back of this slide. So how are the instructions that cells follow written? So we can see in the next slide. Yeah, DNA is the genetic language. The instructions that all life has to follow to be able to exist is written through the DNA. And it's a common language that all cells have, as those instructions are written in the same way for all cells throughout all organisms. DNA stands for deoxyribonucleic acid, and you can see why we need to shorten it with DNA, because it's a very long word. And DNA only has 4 letters. You can see here, T, C, G, and A. 4 letters are enough to give the great diversity to those instructions that make up life in our planet. So I would like to get us started with a questionnaire, a small poll that I'm going to launch soon. And I would like you to see the organisms that I have in the center of this slide. There's a chimpanzee, a banana, a mouse, and an insect. And now you know they all have cells, and they all have DNA, because those are the instructions that the cells need to follow. But I would like you to think about whether you, as a human, share any DNA with these organisms. So I'm going to send a small poll here, and I think I can do it. Yep. I'm going to launch the first poll, which is, with which of the following animals do you share parts of your DNA? So I think that should be available to you right now. And so please submit your questions in the next couple of minutes. And I can see many of you are already voting, so that's great. We're going to keep the poll open for a couple of more seconds here. So I'm going to end the poll quickly here. So thank you very much for your participation. And as you can see, most of you actually got the right answer. We share DNA with all the organisms on the screen. It's true, we share most of our DNA with chimpanzees, but we actually share DNA with all of these organisms. As a matter of fact, it might surprise you to hear that we share up to 40% of our DNA with bananas. And we definitely share way more than that with mice, and we share that with insects as well. So we share DNA with all organisms. And that's why we all have a common ancestor, and we derive from the same ancestor at some point millions of years ago. And just quickly to tell you that DNA is a double-stranded molecule here, and then in the right you can see it. There are 4 letters that are molecules that that compose it, and the sequence of those adenines, thymines, guanosine, and cytosine makes up gives all the information that our cells need to have to be able to function. And another very important feature of DNA is that it can copy. So we have, since we were little, we had a certain amount of cells in our body, and then those cells need to divide, and so that way our bodies can grow. And the feature of DNA that's so amazing is that it can copy itself. So that each new cell that appears throughout our life has a copy of the instruction that it has to follow to be able to function properly. And so maybe now Matthew will tell us how DNA is organized.
Yeah, so you might be wondering, how is all of this information stored in a cell? We know that cells contain a whole bunch of information, but how does it keep track of what all of that is? So it might be useful to think about this in a similar way as how we organize information into books. So in English, we have a bunch of words that are organized, that have letters, right? And each word contains some kind of idea or information. And we can create sentences by putting these words together, and we can form an entire page. And that page can start to form a story, and we can make a lot of pages and put them together to form a book. And a book can contain massive amounts of information that we can read and use to understand a set of instructions. So in a very similar way, a cell uses those 4 letters in its DNA to form something like a word. And you can begin to put those together to form chains of DNA. And these chains of DNA contain information, much like a page contains. And if we take a bunch of those chains of DNA and we create one long, very long chain, and we bundle it, we can create something like a book. And that's shown on the bottom right here. A bundle of DNA is much like a book, and that it contains a lot of information that's organized in a very specific way. So we can think of cells as containing a bunch of books or a bunch of bundles of DNA that contain all of the information that it needs to grow and develop. Next slide. So you might be wondering, why are we using strawberries? And it will probably come as a big surprise to many people that inside a single cell of a strawberry, there's a lot more DNA than in a single cell in your body. So that's depicted here by the number of books. So strawberries contain a lot more books or a lot more bundles of DNA per cell than humans do. So we can use a strawberry as a really good system to extract DNA because there's just so much of it. So we're going to do that today. So if we want to get started, we're going to extract the DNA from a strawberry, and by the end, we should be able to visualize these books or these bundles of DNA in a long strand.
Great, thank you, Matthew. So Gabriela, you can tell us what we will need today to do the extraction. In the meantime, I'll get set up to do it with you.
Great. Yeah, so today, everyone, we are going to do, like we said, a DNA extraction with strawberries. And if you can follow along with us, that is great. These are the materials that you need. You need the actual DNA containing material, the strawberries, a blender. If you don't have a blender, a plastic bag is perfectly good. Water, liquid dish soap, salt, baking soda, a glass, a coffee filter, and rubbing alcohol. And a really important step here is to put the rubbing alcohol in the freezer before we start. It's going to help us a lot at the very end. So Enrico, do you want me to spotlight you?
Actually, before we do that, maybe we can send out a questionnaire to see if everybody has the material they need to do today's activity. And so actually I will send out a small poll right now, which is, do you have everything you need? And this meeting is going to be recorded, so if you want to do it at a later time, you can always find the material, the recording online. And so you can do it at a later time. So I can see a lot of you are responding. Seems like a decent number is voting for yes. Most of you are probably not going to follow along today, but a good number of you seem to have everything ready to do it with us. And so we will have some more poll questions throughout the presentation to make sure that you guys are able to do some of the steps. And so I think we'll end the poll. Seems like about 4 of you will be doing the procedure with us, which is great. That's exactly what we want. And so now Gabriela can tell us what's the first step.
Yeah, so for you going along, the very first step is to make the extraction mixture, which will require 2 teaspoons of dish soap, a teaspoon of salt, all dissolved in 100 mils of water. So just a cup or 2 of water is all you need. And Enrico right now is putting that together for us.
And I'm ready.
Okay, you're ready. Let me spotlight you.
Great, thank you. Great, perfect. Okay, so I hope you can all see what I'm doing here. We're going to start by adding regular dish soap, palm leaf dish soap, and we're going to add it to a container, an empty container. And actually, I think there was a typo on the slide. It's supposed to be 2 tablespoons of dish soap, so I'll add 2 tablespoons of dish soap. So here I have a measuring spoon, and here it is. 1. And 2 tablespoons of dish soap. You can eyeball it as well, it's fine. So now we're going to add salt. So we're going to add 1 teaspoon. Now it's actually a teaspoon of salt. And here we go. 1 teaspoon of salt. And now we're going to add water because we need to dissolve all of these materials. And I have a US cup. And please don't forget to ask your questions through the Q&A, and one of us is going to answer them shortly. So here we have 1 US cup, and I'm going to fill it with regular water and add it in. And then another US cup. Perfect. So that's all we need for the extraction mixture. That's what's going to allow us to extract our DNA. And we have to stir it for a couple of minutes. And in the meantime, maybe we can launch another poll asking whether you have been able to do this. I think we have that poll, right? That's the next poll. Okay, that's actually not the next poll, I'm sorry. So we'll continue mixing for a couple of minutes, and maybe in the meantime, Gabriela can tell us what's the next step. You're muted, Gabriela.
Thanks, Enrico. Yeah, so the next step is to smash the strawberries. So right now, we want to get into the DNA that's hiding in the cells. So the very first step is, of course, just making the strawberries as mushy as we possibly can. So Enrico will bring us through that next step. But this is a step where you can blend the strawberries. If you can't blend them, you'll have you can use a plastic bag, and Enrico will happily demonstrate how he can explain how to do that.
So right now, I think we're ready to move on. We have, and so maybe we can sharing. Yep. Perfect. Right now we have a homogeneous mixture, so you cannot see the salt, you cannot see everything, it's all dissolved. And so we're gonna set this aside, we're gonna use it later. And we're gonna take the blender, or as she said, a plastic bag if necessary, and our strawberry. So we get started with the actual extraction. We need to remove the leaves from the strawberry because we're not gonna need those. So we remove the leaves. And then kids, if you're doing this at home, please let your parents help you with this. You're gonna take a knife and you're gonna cut up the strawberries. And the reason why we're cutting them up is no other than this, the blender will do its job better. It's gonna be easier for the blender to blend the strawberries if they're cut up. So I'm gonna cut them. And I'm gonna cut the next one. Yeah. They're all cut up into pieces. And I'm gonna add a little bit of water, just a tiny bit of water, because otherwise the blender is not gonna be able to blend properly. So that's also gonna happen. I think that's enough water, just a little bit. And now I'm gonna go to the back where I have the motor for the blender, and I'm gonna start blending the strawberries. For cordiality, I'm gonna put myself in mute so you don't hear the loud noise of the blender. And maybe Gabriella can talk about the next steps, and we can just wait a minute and give you time to do this at home. So I'm gonna mute myself. I'm not muted yet.
Okay. Yeah, so while Enrico is pulverizing these strawberries, I'm going to talk to you guys about why we are doing these steps. And so to understand all these steps that we're going through, it's really important to understand what the cell is made of. And so like we've been talking about, and what we're really interested in is the DNA, the master instructions that tell the cell what to do. But the cell is, of course, not just made up of DNA, it's made up of other components. And there are a couple other components that most cells have in common. One are the building blocks that make up the cell, which are called proteins. So proteins are really present in the foods we eat, especially things like meat and whole grains and cheeses. And alongside the proteins are fats. So the cell is made up of a lot of fat that creates envelopes that we'll talk about later. And these fats are just things that we see in our everyday life, like the butter and oil in our food. And so the steps we're going through, we're trying to separate out the DNA from all the building blocks and all of the fats. And let's see. So we'll just wait one moment for Enrico to return. Oh, there he is.
Yep, I am done. So now I'm going to show you how it's supposed to look like. Let's see, maybe we can stop sharing. Yep, perfect. So I think you can all see me. And here is, obviously this looks very much like a strawberry smoothie. It's just water and the strawberry, and they're all the major structures of the strawberry have been destroyed and triturated, and so now we have this, and we can pour it. We can do as Gabriella was telling us. We're going to take 1 teaspoon of baking soda, regular baking soda, and here it is, regular baking soda. The very first thing you're going to notice, and we have to stir it for a couple of minutes. The first thing you're going to notice when you add the baking soda is there's going to be a lot of bubbling happening, and I'll show you here in a second. You can notice that it's kind of, there's a whitish, all those are bubbles. I hope you can see it at home. All these are bubbles that are forming in the strawberry blend mixture. And the reason why that's happening is because the baking soda is doing exactly what Gabriella was talking about earlier. It's going after those little workers, the little proteins that are reading the DNA library in each of the cells. And since we don't want those there because they're going to make it harder for us to extract the DNA, we add the baking soda, and the baking soda neutralizes, goes after them, and stops their function. And so the DNA is not being attacked by or read by these little proteins. And so the bubbling is going to continue for a couple of minutes. And what you want to do is you want to continue stirring all the way until some of the bubbling begins to end. And you can see that the liquid is going to turn darker. Before it was a very light kind of reddish color, but as you will see, and you perhaps can see it right now, it's starting to become a little darker, actually much darker. And the bubbling is going to continue at the top, but it's going to stop towards the bottom of the glass. So we're just going to continue like that. And I think, Gabriella, you were already presenting about the next slide about the several parts of the cell, right? You already shared that.
Yes, yes, we talked
Okay,
about the DNA, the proteins, and the lipids.
perfect. So all that's left to do is for us to continue.
Yes.
And yeah, what's the next step after we add the baking soda?
So after the baking soda, let me get back into share mode. There we go. So we've added the baking soda. So now it's time to, once again, to finally do the last step, which is adding the extraction buffer. And so even though we've done all this work with mashing up the strawberries, the DNA is still inside the cells. So what Enrico is going to do next, he's going to add the detergent to help break down those cells to finally release the DNA from this mixture. And so Enrico, do you want to show that step first before we explain?
We're ready.
Great.
Perfect. Okay, so I'm gonna spotlight myself so you can see me better. And here I am. Okay, so here is the liquid. There is no more bubbling towards the bottom, and you can tell that it's darker. Hopefully you can see with the light that it's darker on the bottom. And so that's what we want. Now we're ready to add the extraction buffer, extraction mixture that we did earlier. And we're gonna add it to equal parts. So there is this much volume of strawberry blend. We're gonna add as much volume of extraction mixture. I think that's about it. Yeah. And now that this is added, we're gonna stir it, just like before. As we stir it, maybe Matthew can talk about why is it that we are adding the extraction mixture, and what's it accomplishing? Stop spotlighting myself so you can see. Okay.
Yeah, so you might wonder why we just added soap. That's a rather strange ingredient. So you can think of DNA as a king in a castle. So it's the master molecule within a cell, and it is contained inside of the cell in a structure known as the nucleus, which is like the castle. So if we want to access the DNA, we have to break down the walls of a castle. So around a cell, there is a membrane, and it's formed with fats. And so the soap that we add helps to break down that fat, much like the soap that you add to a pan when you scrub a greasy pan will help to break down that fat. So the soap that we're adding right now is going to help dissolve the membrane around a cell and allow us to access that DNA.
Yep, that is correct. And so at this point, I've been stirring for some seconds now. And I'll just give it a couple more stirs so that it's all well mixed. Yeah, and I think we're ready to move on. So what's the next step, Gabriela?
Okay. Yeah, so the next step is to actually start separating out the components to help us get to the DNA. So this is our filtration step. So get your coffee filter and a glass to help pour the mixture in to separate out the liquidy part with the DNA and take out all the extra pulp to help us at the end get a lot of DNA out of these strawberries. And so Enrico is going to show us that right now.
Already, perfect. And so I'm going to spell out myself, and here we are. So I've been stirring the soap with the strawberry blend for quite some time now, and it looks pretty homogeneous. It looks pretty well distributed. So now I'm going to go ahead and do what Gabriela was talking about. I'm going to drain the big chunks of strawberry that are still left in the blending into this small glass here. And what I'm using right now is just a regular drainer that you could find in the kitchen sink. And I think this is fine enough so that I don't really need to put some paper because I think it will be able to do the job without the paper. But if you have a more coarse, a less fine drainer, you can use the coffee filter as Gabriela was talking about. Anything that does the job. Basically, we're trying to get rid of the big chunks of strawberry that are still left in the previous steps. So I have it here, and I'm gonna add the mixture. Just like that. Yeah. And you can see all these chunks of strawberry are left on the drainer, and that's what we don't want this. So we can set it aside and we're not going to use it anymore. So this one, we have this more liquid, more pure blend of strawberry that should have the DNA floating inside of it. It should be dissolved inside this blend. And we're ready to extract it. So how do we do that, Gabriela?
Yeah, so this is actually, we're getting to the really fun part now. So now we want to get the DNA to separate out from the water. And we do that by using our chilled ethanol or rubbing alcohol that we put in the fridge at the beginning of the activity. And we'll pour it in nice and smoothly. So Enrico's going to show you, it's really important to be nice and smooth when you pour it in to help us separate out that DNA from the water.
Yep, and we are ready to go move on. Yeah. Perfect. So as you were talking, I went to the freezer and I got the chilled rubbing alcohol. Now it's cold and we need it cold. And I have the solution with the strawberry blend, and I also have a test tube over here. So you don't need to have a test tube. You can have, you know, a small kind of narrow glass container, anything that can hold liquid will do the job. So I'm going to add some of this strawberry blend into the test tube very carefully. I'm going to try not to get the sort of bubbles that are on the top. You may not be able to avoid it. I think it's going to be fine. Just try not to make a lot of bubbles when you pour the strawberry blend into the test tube. So I'm going to do it slowly. And it's working. There. So they get some bubbles, and you probably will not be able to avoid it either, but I have the strawberry blend inside the test tube. Now what I'm going to do, I'm going to take the chilled rubbing alcohol. I'm going to wait a little bit for these bubbles to disappear. Now I'm going to add the rubbing alcohol. And as Gabriela was talking about, you have to be very gentle on this step. You have to be very careful because alcohol can mix, if you do it so vigorously, it can mix the strawberry blend. So you gotta make sure that as the rubbing alcohol goes down, it goes down against the wall of the test tube or the glass or whatever container you're using. If you do it that way, then it will just kind of settle on the top of the strawberry blend and it will not mix. And that's exactly what you want. You want two different sort of layers. So very slowly and carefully, I am adding the alcohol. Like so. And that is it. So I've added 2 layers. You can see probably in contrast, you can see there is 1 layer of alcohol and then there is 1 layer of strawberry. And so I'm just going to let it sit there. And as this happens, you should be able to start seeing there's kind of a cloudy, sort of foggy mist coming out of the strawberry blend. And that, believe it or not, is a DNA that's coming out from the strawberry blend. It was dissolved in the strawberry in the red part of this test tube. And now as it encounters the alcohol, it's aggregating. It's sort of becoming into sort of chunks, cloudy chunks of DNA. And that's the end of our procedure. So we'll just give it a couple more seconds so it happens. And it might be less evident and it's going to be kind of hard to show on camera, but I can already see some of these cloudy sort of mists of DNA chunks that are forming. So I can already see it. And I'm going to give it a couple more seconds so that more of it comes out of the solution. Yeah. So I'm going to hopefully shake it so I can kind of start to come up. And I have a hook here. It's just a paper clip that I modified to be a hook. And I'm going to use it. And there it is. Actually, I think you can see it right now. You see that white cloudy thing that's sort of floating like that? Honestly, it looks like spit, right? But it's not, it's DNA. This cloudy white thing that you see floating, that is the DNA from the strawberry. And it's actually pretty visible, and now it has started to float, and I'm gonna try to grab some of it to show you up close. The problem with grabbing it is that it's very mushy and very viscous, and so it's not easy to actually grab to show you. And I'm having a hard time to see it myself. Some might not be able to do it just because the way it came out of solution. And there's a little bit of a white background making it harder for me to actually see it. And I might not be able to grab it, guys. Yeah, it doesn't always come out in a way that allows you to grab it. In this case, I think it came out in like this chunks, so you can kind of see there are all these chunks floating, these white chunks. Can you guys see it? Matthew, perhaps? Yeah, you can see it. Yeah. So those white chunks, which are too small for me to grab this hook, that's the DNA that came out of the strawberry blend. Sometimes it comes out as a very long thread, and then it's easy to just grab it with a hook. Right now, it came out this way. There's nothing I can do about it, so I can't actually grab it out to show you. But that right there, hopefully you can all see it if you're doing it at home. That is DNA coming out of the solution, and you can see it by yourselves. Strawberries had DNA, and this procedure is now here. So with this, we have come to the conclusion of our activity, and I hope you got some of the results that we got doing at home. Sometimes, you could have gotten better results. I hope that's the case, and I hope you'll be able to see it by yourself at home. So with this, I think we can continue sharing slides and go into a small debrief and recapping what we have done today. Yeah, and I am back up close. Perfect. So what have we learned today? What is the basic unit of life? And so hopefully in the chat right now, you can start asking that answering that question. What is the basic unit of life? What do you think from today's presentation? What do you think is the basic unit of life that makes up all organisms around us? So we'll give you a couple of minutes, and I can already see some answers coming out. So already one attendee has said that the unit of life is a cell, and that's right. Cells are what all organisms share, and they make up our bodies. So they're these small little compartments where everything happens, and they're the smallest unit of life, and we're all made up of cells. So now the next question, as you can see there, is what do all organisms share in their cells? What do all organisms have that carries information about them and gives information about how the cells behave and how then consequently our bodies behave? What is it that all organisms have and that carries information about us? Yeah, and already some people are answering the right answer. So what do you think it is, Gabriela?
I have a little hunch it might be DNA.
Yeah, you're right. It is DNA. And as you can see, it's part of all the organisms around us. And it's not a foreign molecule. It's something that we encounter on a day-to-day. Today we saw it. It's in our food. It's in the organisms that are alive around us. It's in our own cells. So DNA is not a foreign molecule. And it gives shape. It's what informs the decision that cells make and give shape to all the diversity of life on the planet. And finally, this is kind of a trickier question. What barriers had to be broken for us to extract the DNA from the strawberry cells? And Matthew was answering this question earlier on. And so maybe I can give you a couple of minutes now to reflect on what did we have to do to actually be able to reach the DNA inside the cells. And I already see somebody is giving good answers of what has to be broken to be able to get to the DNA. And yeah, maybe Matthew, you want to recap?
Sure. So cells are all surrounded by a membrane that's composed of this fat, like it's called a fatty lipid or a lipid bilayer. And so you have to break through that with a detergent in this case to access the DNA. And then inside of many cells, there's another structure called the nucleus, which has another membrane that has to be broken to get to the DNA inside the nucleus. That's the king in the castle.
Yep, that's perfect. And so I can already see several questions. And there are some questions that we may have not been able to get to before. Somebody was asking, why do strawberries have so much DNA? And Matthew was already talking about this. All organisms have different amounts of DNA in their cells. They have like kind of libraries. You can think of them as libraries. And strawberries happen to have a way bigger library than other organisms. They have gone through some events in their evolution that has led to them having a lot of DNA in their in their cells. And actually, you can see that the wild strawberries are much smaller because they have less DNA. But actually, the strawberries that we eat on a regular basis, they have a lot more DNA, and that's why we select for them, because they have a lot of DNA and are much bigger. Somebody's asking now if strawberry seeds contain DNA, and what do you think, Gabriela? Do strawberry seeds contain DNA?
All seeds do carry a copy of DNA. So DNA, one of the central parts of being alive is being able to pass on your DNA to your offspring. So all those seeds have the first instruction set that will tell the seed how to sense the water and light and how to start growing. It's a really, really impressive system.
Perfect. And so we're going to wait for other questions to come. And I think somebody already answered one of the questions that I was seeing. Yeah, perfect. So Matthew already answered one of the questions written there. And what does DNA stand for? So you may remember from one of the earlier slides, DNA stands for a very long set of words, and that's why we use, an acronym. It's deoxyribonucleic acid. So you can see it's a very long word, and it has to do with how DNA is chemically composed. And that's, the word deoxyribonucleic acid has information that chemists and biologists can recognize that tells them exactly what molecules make up that bigger DNA molecule. And so maybe someone else wants to answer one of the anonymous questions here. What is the difference between RNA and DNA?
So this is a question I really like. So DNA and RNA are similar in the fact that they have a similar structure and they are string-like. But DNA is made up of 4 types of letters, and so is RNA. It's made up 4 types of letters, but one of the letters is different in RNA. So it's a slightly different instruction set. And the role of RNA in general in biological organisms, they help make tiny copies of specific parts of DNA that's nice and safe in the middle of the cell, and they take it out to the factories in the outer parts of the cell to help start building the building blocks. So the RNA is actually the step in between DNA to protein, making the building blocks.
Perfect. And so someone else is asking whether strawberries have more genes, longer genes, and non-coding DNA. So those are really great questions. And actually some of these answers are complex. And so I'll try to keep that answer short. So strawberries may have more genes. So there's a repository of information about how many genes are in strawberry and how many genes are in any organism that's online. And scientists use these repositories of information to be able to know how many genes are in an organism. Strawberries, I would suspect, may have more genes. They don't necessarily need to have longer genes. The length of a gene depends on what that gene is trying to code for, what information, what building block that gene will create. So a longer gene will probably create a longer building block, and a shorter gene will probably create a shorter building block. And so it's not necessarily that strawberries will have longer genes. They might, but they don't have to. And they also contain non-coding DNA, and non-coding DNA is something that is being studied actively for the functions of it. And it's hard to answer whether strawberries contain more non-coding DNA. For that, you would actually have to go and look up the DNA sequence and use some special programs that scientists have at their availability, and you'll be able to see exactly which parts of the DNA strand code for building blocks, code for protein, and which parts of the DNA do not code for protein. And then you'll be able to get an answer. Right now, those numbers are so hard to remember that at this moment, I do not really know, but there's not necessarily a correlation with DNA having with strawberries having more DNA and also having non-coding DNA.
And I do want to add on to that. So plants, for a reason that we scientists are trying to figure out, tend to have many, many copies of DNA. And for some reason, it's really easy for them to double the amount of DNA and create new species. And so that's something that we are trying to figure out. So like Matthew was mentioning earlier, in our cells, we have 2 copies of the instructions. But in strawberries, they have 8. I think wheat has 7, which is an odd number, but it's really a really fascinating characteristics that plants tend to have many copies. So that's a huge question in plant biology right now.
Maybe somebody can answer, what does RNA stand for? Matthew, you want to answer that?
Oh, I was going to do the next one, but I can do this one. So
Oh.
yeah, RNA, so DNA is deoxyribonucleic acid. RNA is just ribonucleic acid. So they're very similar, and that just is talking about one of the parts of the molecule. They're very similar. There's a slight structural difference, and so that's what the R versus the D stands for. But I wanted to answer actually the one below it. So how are we similar to bananas? And this is a really good question, because you might look at a banana and think there's nothing that we could possibly share in common with a plant. But that's not actually true. So if you think about what all cells need to do, so all cells need to grow and divide, right? And in order to make a new copy of a cell, you have to copy your DNA. And that requires little molecular machines that will actually go in and copy the DNA. And so in a banana, you'll have to have those machines to copy banana cells. And in your body, you have to have little machines that will copy your DNA. In the same way, a banana cell needs to get energy from sugars, and our cells need to get energy from sugars. And so we have very similar components for some of these very fundamental things that all cells share, and that makes us all share DNA. But we also have many differences, and that results in many different aspects of our DNA. But we have a pretty standard core that all forms of life share. So that's why we all have, even with bacteria, we share some DNA.
Perfect. Thank you very much. And the next question is very interesting. Maybe somebody else wants to answer it. So why do we need to know about strawberry DNA? And Gabriela, do you want to answer that?
Yeah, so there are so many reasons. I could go on and on about why we're interested in DNA of a particular species. So one of the main like lines of thought or reasons we study DNA in plants is to understand like how they react to light, how do they grow, how do they react to things that are trying to eat them, how do they protect themselves when there are lots of strong salts around them. So organisms have to, especially plants, have to adapt to lots of changes in our environment. And all that responses, all the ways they work, all the ways they grow and can divide, that's all encoded in their DNA. So lots of questions we have about life are in DNA, which is why biologists are always studying DNA. That's why I extract DNA from my samples every day and trying to understand what that code is trying to tell us in the long run. In particular in strawberries, for strawberry research, we probably want to understand what genes help make the strawberries sweeter, what genes help the strawberries grow bigger. And if we can understand that, we're able to grow better strawberries. So there's just so much you can do with the information DNA contains. It's really cool, and that's why I'm here studying biology, particularly in plants.
Yeah, and I would just like to add that ultimately, the information that a strawberry or any other plant has in their DNA, especially for crop plants, like the ones that humans depend to survive on, is important information that if we can study it, we can help farmers make better decisions about how to grow those crops. We can perhaps find better ways to grow those crops. We can find some situations where those crops would suffer or be better off. And if we can have that information, then farmers can grow better crops and feed more people with them. And that is something that has always been important for humanity and will continue to be important as our planet goes through some important changes. And we'll need to have more food for more people in a harsher environment. So that is exactly why plant biologists are necessary and why we try to understand why how plants behave and how plants grow and how plants react to their environment, because they're so important to our survival, not just our survival, the survival of everything in the planet, but also ours. So that is the last. Oh no, there are more questions.
I can get the next one. So the next question is, what are genes? And this is a really good question. We've been talking about genes without really explaining what we mean. So if we think back to the slide that I shared where we had a bunch of words that then form sentences, that then form a page, that then form a book, we could think of genes as a kind of a sentence. So it's a sequence of DNA that has some kind of information. And when we read a sentence, we read from the start to the end and we understand what that says, and then we can interpret that in our mind. And in the same way, we have sequences of DNA within a cell that can be read by the cell to confer a piece of information, and that will allow the cell to respond to whatever that piece of information is. So you could think of a gene as a piece of DNA that has a functional unit of information that can be read. And so it's like a sentence of DNA that can be read by the cell, in many cases to produce like a little machine that can be used to perform a function within the cell. It's a really good question.
The next question is a hypothetical, and what would we be like without DNA? Well, that is something that there are some fields in biology that study the question of how was life before DNA formed. DNA formed millions and millions and millions of years ago. And some people think that before DNA, there were molecules in sort of this prehistoric, very ancient blob of liquids in an earth that was much different from how we have it right now, where there were a lot of volcanic explosions and a lot of elements all floating around. And some very special circumstances, which are still being studied, led to the development of some molecules, some chemical reactions that then continued on, giving rise to more chemicals and more reactions that eventually developed into, scientists believe, RNA organisms, RNA structures that, if you remember, RNA is the other nucleic acid. And then from there, something happened that eventually led to organizations of molecules with nucleic acids inside, either RNA or DNA. That question is very much still debated. But to answer what would be like without DNA, well, we probably wouldn't be at all. So much of us and so much of all the organisms that we know, it's determined by DNA. So without DNA, life as we know it, all the life as we know it, wouldn't exist. Not even viruses, because viruses, some of them are RNA viruses, but they still depend on DNA structures, on living cells that have DNA inside of them. So none of the life that exists currently would be possible without DNA. And talking about DNA, what is the most fascinating thing about DNA in your opinion? So now this is a very subjective question. So maybe some of the other volunteers can talk about this. Something that I think I thought about long ago. Now I've forgotten why, and I have forgotten an answer to that question. So maybe the others can talk about it.
I think what I love about DNA is the fact that I think that what is really cool about DNA is that we share it across all life. So what we were talking about earlier, we share a lot of the same genes, as Matthew explained earlier, with bananas. Right. So the fact that DNA is so versatile, and throughout the millions of years that DNA has been fueling the process of life on our planet, it has been really creative. And so seeing and studying the diversity of DNA and what it can do and how it's changing and how it continues to change, DNA will continue to change throughout time. We're not at the end point, right? So such a versatile, fascinating molecule to study.
Yeah, I would like to build on that. So the process of evolution that we all have heard about relies on DNA and how DNA slowly changes over time. So ideally, when a cell copies itself, it wants to make a perfect copy. But sometimes a mistake is made, and those mistakes over time will begin to build up, and you get different DNA. And over thousands of generations, and tens of thousands or hundreds of thousands of years, you can actually begin to create new species. And those new species are derived from changes in DNA. So for me, the most fascinating part of DNA is both that, again, we all do share DNA from the strawberry that we just saw extracted. We just saw DNA extracted, and that same molecule is found within your body, and it's found in bacteria, and it's found in fungi, and in plants, and in animals. So that same molecule is found in all life, and as it changes over time, we can create new species and new different forms of life. And that's a really exciting idea that you can watch over time if you look back into what the sequences were, how things changed and became adapted to different environments over time.
And I will share my opinion on this. I think all of those reasons that Matthew and Gabriela were talking about are fascinating. To me, the most fascinating part of DNA, it's how it's complex and simple at the same time. Because remember what we were saying before, there are only 4 letters, 4 parts of DNA that are distinct from one another that make up DNA. And out of those only 4 letters, you can get so much information in one single cell. And then across organisms, DNA can be so diverse to give so many different shapes and forms to life the way it is. And to me, that's fascinating. Imagine if all the books that humanity had ever written had been written with only 4 letters. You imagine how fantastic, being able to express so much with only 4, components, 4 letters, 4 molecules. That is, to me, very, very fascinating. And I think the next question is, do we share DNA with all the organisms on Earth? And this actually goes well with a comment somebody had earlier on the presentation. Things that were commenting that, oh, well, no, we don't share DNA with bananas or chimpanzees because they're different species. And the answer is, yeah, we share a percentage of our DNA with all the organisms on Earth, all of them. And what matters is how much of our DNA we share. So we share quite a bit of our DNA with chimpanzees. You can see that chimpanzees, the cells of chimpanzees, that information that tell the cells how to make an arm or how to make eyes. Both chimpanzees and us have eyes and arms. And so the information for those parts is to a great extent similar. Not 100% similar, otherwise we would be chimpanzees, but very similar to a great extent. And we share information with the banana. As Matthew was saying, there are some things that banana cells and human cells need to do. And for those processes that are similar, the information is very similar as well. So that's why we share information with those organisms. Yeah. And there is a next question, which is kind of a hot topic. Somebody else wants to go first and answering this question, or trying to answer this question? It's a very controversial kind of topic nowadays.
Yeah, so I'll start off, and if you guys have anything to add to it, please do. So GMO foods are foods that have been have a trait change based on a genetic modification. So scientists go in, they find a gene that maybe if you alter the gene, you can make the corn bigger, you can make the fruit bigger, things like that. So GMOs have been purposefully changed and bred to look the way they do. And GMOs, so the technology to do that is really incredible technology, and the benefits we've had from it in the United States have been like insurmountable economically. So the concerns around GMOs, from what I hear from friends and family who don't understand it, they think that for some reason it will make them sick. In general, though, most of that fear stems from misunderstanding and not knowing how DNA works, right? So that's why we're doing an activity like this with you, showing that there is a lot of DNAs in every organism, and we eat it all the time. We're eating DNA all the time. It's not strange to be eating it. And also it stems from the fact that, and DNA is not, the rules that we have in our cells, they're not going to stay the same forever. Evolution occurs because DNA changes. And so I think some people don't understand the fact that DNA is always changing. I think the fact that they hear that it's being modified sounds scary and sounds like we're messing with something that shouldn't be messed with, which just isn't the case. DNA is always changing nature. Organisms actually switch DNA between each other, which is a really cool process. And so I think it's always smart to question things that you're being given and being informed about things you're eating. But in this case, GMOs are a phenomenal technology, and all the GMOs on the market have been proven to be safe in thousands and thousands of really well done tests.
I want to get to, that's a great answer. And just though to link it to the one below it. So does DNA sometimes cause cancer? So the molecule of DNA itself does not cause cancer, but the way that your DNA in your body is read can sometimes cause cancer. So you think about if you're typing a Word document, writing sentences, sometimes you might misspell a word, right? And you cause a typo. And in the same way in your body, sometimes as your DNA is being copied to make new cells, a typo will form and you'll get a little mistake. And your body can then read that mistake in an improper way and cause cells to grow out of control, and that will cause cancer. But the molecule of DNA itself does not. So you can eat DNA and it won't cause cancer, but the DNA that forms your body sometimes will change naturally over time when you're exposed to sunlight or just by chance. And there's this very, very, very small chance, but a possible chance that over time you might develop cancer. But it's not from the DNA being toxic, it's just from the sequence of DNA.
Exactly. And so that's why there are some organisms, some chemicals that are dangerous, because they, when in contact with your body, contact with your cells, can go inside, can penetrate through the cell and go to your DNA, not the DNA you've been eating, your DNA, and change it in a way that then leads to that cell to grow out of control. And that's eventually resulting in a tumor. And although eating DNA is not dangerous, I would like to take this chance to tell you, please do not eat this. After we have done it, do not eat this, do not eat any of these, not because there is DNA, but because there is detergent and alcohol. So I just want to disclaimer there, please do not eat. Yeah. Okay, so I think we're about out of time. Do we consume DNA? Will we actually break down and absorb any of it? So I'll answer this last question. We consume DNA, and just like with anything else we consume goes through our stomach or digestive system. Our digestive system indiscriminately breaks down everything that is in our food, and that includes DNA. And those molecules that make up DNA will eventually be absorbed by our body and be used as fuel, and some of them will be used for other purposes. At the end, that's a complex topic. We can answer it very briefly here. The food that goes through our body gets broken down no matter what it is, no matter if it's protein, no matter if it's DNA, all of it gets broken down and then absorbed by your body equally, and then has different functions. I think that's all the time we have with you today. Thank you so much for joining. I hope you enjoyed today's presentation, and it was a pleasure for us to be with you. And I would like to thank all the volunteers that participated today for making this activity possible, and the Bay Area Science Festival for giving us a platform. Thank you very much.
Yeah, thank you so much.
Thank you, everyone.